so stressed about DNA replication. Mechanism of DNA replication, semi-conservative, IN blur.
Oh, Xiaomi, you're so lucky because Nabila and I have learned about this topic. We can explain it to you.
Yes, thank you.
Okay, in this video, there are 3 alternative models that you need to know. First, conservative model, semi-conservative model, and dispersive model. Okay, for conservative model, the parental double helix remains intact and an all-new copy is made. Second, semi-conservative model. The two strands of the parental molecule separate and each functions as a template for synthesis of a new complementary strand. Okay, the last model is dispersive model. Each strand of both daughter molecules contain a mixture of old and newly synthesized parts. Okay, this is the mixture. This is a short segment of DNA. This is the parent molecule. This is the separation of strands. And this is the daughter DNA molecules. Each consisting of one parental strand and one new strand. Michelson and Stahl experiment proved that DNA replicates in a semi-conservative manner. Hi guys, in this video I will explain about replication bubble. In eukaryotic cell, they have multiple origins of replication. They're speeding up the copying of the very long DNA molecule. Replication begins at specific sites where the two parental strands separate and form replication bubbles. The bubbles expand laterally as DNA replication proceeds in both directions. Eventually, the replication bubbles fuse and synthesis of the daughter strands is complete. In an E. coli cell that is prokaryotic cell, usually have only one origin of replication on each circular DNA molecule. This is the differences between prokaryotic cell and eukaryotic cell. But the way how two daughter DNA molecules produced still the same. Okay, next, there are 5 enzymes and 1 protein are involved in DNA replication. Okay, first, DNA helicase. Its function is to unwind the double-stranded DNA to become single-stranded. Okay, next, single-strand binding protein, that is SSB protein, function to stabilize and prevent the single-stranded DNA from recoiling in helix until the strands are replicated. For topoisomerase, it helps relieve the strain caused by the unwinding of DNA double-stranded. Next, primase is used to synthesize RNA primer by adding RNA nucleotide that complementary to the DNA template. DNA polymerase III function to add free DNA nucleotides to the 3' end of the short RNA primer. DNA polymerase I will replaces the RNA primer by adding free DNA nucleotides, producing new complete DNA strands. Okay, lastly, DNA ligase will form phosphodiester bond to join Okazaki fragments together at lagging strand. There are 3 processes in DNA replication. First, unwinding of DNA strand. Second, elongation. And the last process is joining Okazaki fragments. Okay, DNA replication begins from origin of replication. Then, enzyme DNA helicase will unwind the double-stranded DNA to become single-stranded. Both single DNA strand will act as template to synthesize new DNA strand. This occurs in both directions in the replication bubbles. When DNA unwinds, Y-shaped structure called replication fork formed. After that, single-strand binding protein, that is SSB protein, will hold the separated DNA strands apart while they act as a template. SSB protein function to stabilize and prevent the single-stranded DNA from recoiling in helix until the strands are replicated. Then, untwisting causes tighter twisting and strain ahead of replication fork. So, enzyme topoisomerase helps relieve the strain caused by the unwinding of DNA double-stranded. Okay, we are done with unwinding of DNA strand. After that, we will go to the next process, that is elongation. Enzyme primase will bind to the DNA template to synthesize RNA primer by adding RNA nucleotide that complementary to the DNA template. RNA primer is the short segment of RNA that have only 5 to 10 nucleotides. After a few nucleotides have been added, the primase is displaced by DNA polymerase III. DNA polymerase III adds free DNA nucleotides to the 3' end of the short RNA primer. The free DNA nucleotides is complementary to the basis of the DNA template strand. The leading strand is new DNA strand synthesized continuously and towards the replication fork. Meanwhile, the lagging strand is new DNA strand synthesized discontinuously and away from the replication fork, forming Okazaki fragments. To make it more clearer, this is the direction of 5' to 3' end. And this is 3' end to 5' end. The elongation is in the direction of 5' end to 3' end, while the DNA template is in the direction 3' end to 5' end. This is how we read the direction, because for leading strand, it is towards the replication fork. Then, we will proceed with our last process, that is joining Okazaki fragments. Okay, soon DNA polymerase I will replace the RNA primer by adding free DNA nucleotides, producing new complete DNA strands. Okay, for lagging strand, the process is still the same. The direction is from 3' end to 5' end, and it will still be the same. Primase will bind to the DNA template, and then DNA polymerase will bind. It will be here. After that, DNA polymerase I will replace the RNA primer by adding free DNA nucleotides, producing new complete DNA strands. DNA ligase form phosphodiester bond to join Okazaki fragments together at lagging strand. Okay, this is the direction of lagging strand. For summary, both parental strands of DNA act as a template. The elongation is in the direction of 5' end to 3' end. And lastly, DNA replication produced two identical copies of the original DNA.
So, Xiaomi, do you understand about this DNA replication?
Yes, I do.
We hope that you guys can understand what we explained.
Bye!